Okay, so welcome back, everybody. Our goal today for this lecture is we're going to finish up our discussion with the anatomy and physiology of the cell. All right, so we pretty much talked about all of the anatomy stuff related to cells. We talked about each of the organelles so far and what each of the organelles do. All right, and then now what we're going to end up talking about is how does this cell work, right? Or how do cells in our bodies work? So one of the big kind of concepts that we should understand after this is cell metabolism. Okay, so how do cells survive? How are they able to function? How do they process nutrients? How do they get rid of waste? Okay, so that's going to be one of our first things here, is we're going to talk a little bit about how these things function. One of the main ways they do that is through this stuff called cell or membrane transport, rather. So the way that we can get things across this semi-permeable barrier is going to be important, right? So that's going to be how we control, or how our cells control everything that comes in and out, is by utilizing different ways to get stuff across this barrier. So that's going to be important. Then the next thing we want to understand is, okay, so now that we can get stuff across this barrier, how do we select for that, right? So selective transport kind of folds in underneath membrane transport here. All right. So once we've gotten things across, then how do we express information that our cell needs? How do we convert our nutrients into usable energy, and how do we respond to that? So then protein synthesis is another concept that we'll end up talking about. And this kind of encompasses two different processes that we call transcription and translation. Okay. So we talk about going from one gene to one protein. That's really what we're talking about. So yeah, I'll kind of rewrite this. So a better way to think about that is something, if we have the word gene to protein, it's a better way of thinking about that. And then how do cells replicate? So mitosis. Okay. So these are kind of our big ideas for today's lecture and for chapter 3. So now that we've talked about all the things that make up the cell, now we've got to figure out, okay, how does the cell operate? How does it do these things or perform these actions? Okay. So this is where we left off last time. We now understand how the cell membrane is put together. We know that the cell membrane is made out of something called a phospholipid. And remember, phospholipids are unique, because they have two different regions on them. Remember from last chapter, in chapter 2, we talked about the molecular structure of each of our 4 macromolecules. And remember, lipids don't like to be around polar environments. And most of the environment inside the cell and outside the cell is polar because it's mostly made out of water. So the cell membrane is composed of a region that likes to be in water, which is where all these blue beads are, and those are going to be the phosphate groups that are attached to the lipid fatty chains that we see in the middle of this sandwich layer. Okay, so the big technical term we use to describe the cell membrane is something called a phospholipid bilayer. All right. So this amphipathic molecule of a phospholipid, meaning that it likes to be in both polar and non-polar environments, is key because we can control moving things in and out of the cell through different mechanisms. So let's talk about some of those. All right. So these are the main ways of molecular movement in cells. These are the major modalities that we'll talk about. So the first one here being diffusion. So diffusion, if you guys recall when we talked about that phrase, concentration gradients, diffusion is the mechanism of movement that describes that. So diffusion is literally with the concentration gradient through the lipid portion of the cell membrane or through membrane channels. So when molecules move about, every time if they move by diffusion, they're always going to go with the concentration gradient. So you always go from a high concentration to a low concentration. So there's some other figures in here that we'll talk about in a sec, but let's say we've got a big collection of molecules up here. So that's what all these little dots are. These are all of our molecules, okay? And then down here, we've only got a couple blue molecules that are all spread out here. So if all of these molecules are allowed to move out in a random order, they're going to move from a high concentration to a low concentration. So they will move with the concentration gradient, or they will move down their own concentration gradient. So always from high to low. Always, always, always. If they are being allowed to move about randomly. That's the key takeaway there. Because sometimes it's in our benefit to make things go from low to high concentrations. And we'll talk about that once we get into, things like neuron function and that sort of stuff. But any molecule we talk about, doesn't matter what it is. If it's a molecule, if it's an ion, if it's an atom, whatever it is, everything in the universe moves from high to low concentration if they're allowed to do so randomly, okay? So this is important because when we think about the random movement of most molecules, or if you want to think of stuff that's dissolved in water, we would use the term diffusion. And the word osmosis literally means the same thing, but we're just changing our frame of reference to not be talking about some sort of a dissolved solute, but rather the movement of the solvent, so the movement of water down its own concentration gradient. And then we've got some figures that'll make that a little bit more clear. But diffusion and osmosis both describe the same phenomenon, moving from high to low concentration. The only difference is that diffusion is usually talking about the stuff that is dissolved in water. So things like oxygen, carbon dioxide, different kinds of ions like chlorine, urea, right? All of these things are good examples of molecules that can be dissolved in water and will move from high to low concentrations. Osmosis is the specific phenomenon of tracking the movement of water down its own concentration gradient. And that matters because of the way that cells will respond to water concentrations, okay? So diffusion and osmosis are what we call, they're kind of under this umbrella term of something that we call passive transport. There's no energy that's involved. So you'll see here, requires ATP? No. None of those require any energy investment at all. So they can happen randomly and do so randomly. But then sometimes we have methods that will require energy. But there are there's kind of another blanket term of movement that we call receptor or carrier mediated transport, okay? So this means that instead of just moving about in random space, sometimes molecules need a specific protein to get across the membrane. So like in this previous figure over here, we talked about this red channel protein, this membrane channel. This would be an example of a protein that participates in facilitated diffusion. So this channel protein provides a pathway for some molecule, whatever it is, for some molecule to move down its own concentration gradient, whatever direction it may be. So when we talk about facilitated diffusion, think of those channel proteins. They act as passageways. So if you've got a bunch of boulders that are rolling downhill and roll into the base of a mountain, all of those boulders are going to stop, right? But if you add a tunnel through that mountain, then all those boulders are going to be able to keep on rolling through, right? So facilitated diffusion is where you have the ability to allow for typically charged particles or large macromolecules like glucose, for example, the ability to move down their own concentration gradient. And importantly, this doesn't require energy. But then most other forms of what we call active transport, which again is under the umbrella of receptor-mediated transport, because you need proteins. So this term carrier, or I'm saying receptor, I'll say carrier because it means the same thing. But carrier-mediated transport is where you have proteins involved. So when we talk about carrier-mediated, think of these things that are embedded within the membrane. And it's not necessarily important, but most of them are what we call transmembrane proteins. So they span both sides of the membrane. So active transport is where we utilize energy to force molecules or ions against their gradient so that we can move them from low to high concentrations. Well, why the hell do we want to do that? Well, this mechanism is the reason we are alive. This is the reason for our mitochondria meme. It's the reason that the mitochondria is the powerhouse of the cell, because we invest a little bit of energy to shove hydrogen ions against their own gradient, just so that we can allow them to come back through another channel protein that we'll talk about later on at the end of the year. But that's the whole premise for a molecular process that we call the electron transport chain, which is what gives our cells a crap ton of energy for a single molecule of glucose. And crap ton is a metric term. You can quote me on that. But the point is active transport requires energy, and it's used to force molecules or ions against their gradients. So going uphill, think of walking uphill. You need energy to walk uphill, but you can kind of fall downhill, right? And like gravity help you that way. So sometimes there are these things called secondary active transport, where you can move molecules or ions against their concentration gradient, and the energy for secondary active transport of one substance comes from the concentration gradient of another. So there's a really popular protein mechanism that we use to explain this, something called the sodium-potassium pump, where we use the concentration gradient of sodium to allow sodium to move down its own concentration gradient to force potassium up its own concentration gradient. Okay. So that's kind of the differentiation of methods of carrier-mediated transport. Okay, so we have one example that doesn't require energy, but these other two, active and secondary active transport, do require energy. Okay. Okay, Raina, no worries. Just try and hopefully you can get it repaired as soon as you can. You should be able to do the lab on your phone if necessary, because it's just a Google form. Okay, and then the last two that we'll talk about, you're welcome. The last two we'll talk about is how does the cell eat things or consume things, and then how does it get rid of stuff, right? Think of it as us eating and pooping, or eating and peeing, right? Or drinking and peeing. So endocytosis is where cells will consume different particles from their environment, whether they're food particles or debris or sometimes viruses. It just kind of depends. Cells can consume things from the external environment, and they move that material into the cell. There's a couple different ways the cell can do this. Sometimes it'll use carriers as a way to do that, just like we have up here in carrier-mediated transport. Sometimes cells will utilize proteins to bring things in. That's called carrier-mediated endocytosis, but sometimes they can just be moved into the cell just by interacting with the cell membrane and being large enough to cause, the technical term is we call it an invagination of the cell membrane, where it kind of folds in on itself. You can think of like kind of Play-Doh that kind of, oh my gosh, sorry, my cat just hopped up. You can think of it as like Play-Doh that ends up being kind of compressed in on itself, right? And then exocytosis is the exact opposite of that, where cells are going to move things outside of itself. And it'll do that through vesicles, through these little collections of phospholipids, and create these little bubbles to move particles out of the cell, right? Okay, so these are the different ways that cells can move things across their membrane, right? Okay, so when we talk about diffusion, this figure is a really good depiction of diffusion, right? So if you put like a sugar cube in water, over time, the particles and the molecules that are associated with that sugar molecule will dissolve into the glass, or in this case, beaker of water. And they go from a high concentration when they're in that cube, because over on the left-hand side here, all the sugar is mostly concentrated in this cube. The rest of this glass of water doesn't contain any sugar molecules at this point. But over time, those molecules will dissolve. And when they do, so that's the act of the water getting in there and the polar molecules of water getting in and forming hydrogen bonds around the molecules of sugar. As water begins to do that, we see this increase in the concentration of sugar around the beaker of water. Once the sugar dissolves, it doesn't just stay in one part of the solution here. It spreads out, it diffuses, okay? So there are some instances where substances won't actually diffuse, and those are called non-homogeneous solutions, all right? But that's a discussion for another time. This, we're seeing that these molecules right here, we're calling them sugar, can move down their own concentration gradient, where they're in high concentration in the sugar, and then or in the cube right here, and then as water acts upon it, those molecules will spread out and eventually take over the entire glass of water, okay? Now, I'm not proud of this, but what I've done before in class is, it's the same thing with smells, right? So if you have like a particular smelly, bodily function, and that happens, the people who are going to smell that first are the unlucky people in the front row of the lecture hall, right? And then everybody in the back is going to end up smelling that last, right? Because all of those molecules are in high concentration up in the front, and then they eventually disperse or diffuse throughout the room over time. It's the same premise for using, diffuser machines to diffuse things like essential oils or those sorts of things, or humidifiers, right? The whole premise is you diffuse these molecules into the air, and they move from high concentrations to low concentrations, okay? So this figure shows us a good example of how our cell membranes can be selective in what they allow to pass across them, okay? So for the most part, our cells will allow relatively small lipid-soluble molecules to pass across it without any help. So on the right-hand side over here, anything that can dissolve in fat can get across our cell membranes pretty easily. Because remember, the inside of our cell membranes right here, this is all made of fat. And the cool thing about chemistry is that stuff that's made out of the same things as other things will end up dissolving or attaching to it, right? So lipid soluble or fat soluble molecules can pass through the membrane pretty easily and don't need any extra help for the most part. If they're pretty big, and that's when they'll typically use a protein, and depending on the scenario, they might need some sort of energy to be able to be moved across the cell membrane. Certain kinds of hormones that are lipid soluble but are just too big will operate this way and get into target cells that way. And then we have non-lipid soluble molecules that can't get past the fatty acid chain in the middle here. So the non-lipid soluble molecules need help. They need an ability to get across the membrane. And these non-lipid soluble molecules will typically need a transport protein in order to do so. And then there's these non-soluble molecules or ions that can be specified with a given protein that can use those proteins as channel proteins. And if we're thinking about, kind of integrating our understanding of that, channel proteins, again, would be an example of something like facilitated diffusion. But then sometimes if you are forcing molecules against their gradient, then you would need to have proteins that only open one way, and that would be an example of something like active transport. But that's not depicted here. So our membrane channel right here is a really good example of how we can have selective barriers for ions and molecules that are specific to this particular protein. So the cool thing here is, since it's color-coded, it kind of works out. But these green molecules would need a green protein to get across. These red ones match up with this red protein so they can get across super easily, right? So this depicts the fact that our cell membrane is semi-permeable. It lets certain things across and doesn't let other things across unless there's a specific protein to allow that to happen. So selectively permeable, or sometimes we'll call it semi-permeable. Okay. So here's a really good example of how we can also have a selective amount of molecule movement. But this one's kind of interesting because it factors in the charge that's associated with ions like potassium or sodium or chlorine. So we'll talk about this more a little bit later, but remember that some of these elements are ions and they have charges associated with them. So over time, what will eventually happen is certain sides of the membrane will develop a polarity. They'll develop a charge that's associated with them. So not only will there be a chemical gradient, but there will be an electrical gradient that's involved. That's something that we call an electrochemical gradient, and that's an important phenomenon for describing how things like muscles create muscle contractions or neurons create action potentials, or I shouldn't say create, they generate those things, right? And they're sensitive, the membranes of muscle cells and nerve cells, and most of the other cells in our body too, are sensitive to the charges that are on one side of the membrane versus the other. So for example, what we're seeing here is in purple, we have a potassium channel that only allows specific ions to pass through, in this case, potassium. And they can only go one way. They can only go down their concentration gradient, right? Excuse me. But then over here on this pink protein, this pink sodium channel here is what we call a gated channel. And it needs energy to open up, right? So if you add some phosphate molecules from ATP, which is what we're seeing here with these little yellow spheres, those activate and open the sodium channel. So the other cool thing that we can do with these proteins is we can turn them on and off to allow certain ions or molecules to pass through. So when the sodium channel is closed, if it's not activated, if we don't utilize ATP here, then sodium can't get across. But if we open it up, then sodium is allowed to get across, okay? All right. So this figure highlights the phenomenon of osmosis, okay? So diffusion in the last figure talked about the random movement of a solute or a dissolved substance within a liquid, okay? So osmosis, remember, specifically we're talking about, we're changing our frame of reference to the thing that's doing the dissolving, in which most cases in anatomy is going to be water, right? So what we're seeing here at the top, all right, we've got this little beaker that has all these different dissolved solutes in here, okay? So these green and red molecules within this solution, there's relatively little water content in this beaker at this point. But what we have on the bottom is we have a little membrane that only lets water molecules through. It doesn't let these larger dissolved salts pass through. Only water can move across this barrier. It can go into the beaker and it can go out of the beaker, but that's important. So what we'll see is that over time, that if we submerge this beaker with this open bottom that's only permeable to water, is that over time, the volume of the beaker goes up, all right? And it might be kind of hard to tell, but if you have your book next to you, you can see the coloration of the solution gets lighter as we move from left to right. So not only is the volume increasing because water is moving down its own concentration gradient. So if we think about it, with this figure up here, we can see that in this larger beaker, it's all water, right? So you would say that the concentration of water is high in that area compared to inside the beaker where water concentration is low. Water is just like any other molecule. It wants to move down its own concentration gradient. And if you let it do that, then it'll do that. And that's the phenomenon of osmosis. So over time, we can see water will move into this beaker, increasing the volume of our salt solution and thereby diluting the concentration of salt within that beaker. So when we talk about diluting something, that's what we're doing, is we're making the concentration of whatever salt we have dissolved here relative to the surrounding solution, we're making that less. So it says over here that we start out with a 3% salt solution. If we add a bunch of water to that, that number is going to go down because we're making it less. We're making it less concentrated with salt relative to the volume that it's taking up. So we're adding more water to dilute that solution, okay? So that's osmosis. It's exactly the same thing as diffusion, but we're just thinking about the movement of water instead of whatever salt we have dissolved in it, okay? And that's important for animal cells because unlike plants, animal cells don't have these things called cell walls. Plant cells do really, really well under what we call hypotonic solutions, where the inside of the cells in plants are typically, they have more salts and sugars dissolved into them. So the cell will readily take up a bunch of water, because if we have more salt and stuff inside of whatever object we're talking about, water is going to want to move into there. Okay, so plants like that because they have these cell walls that won't allow the cell to explode. But humans, we don't have cell walls. We just have a cell membrane. So our membranes are only made out of phospholipids, and if the pressure, if the hydrostatic pressure inside gets too much, if you have too much water and it's pushing out on the cell walls, the cells will rupture or they'll lyse as a result of that. Okay, so cell lysation is cells exploding. It's just a fancy word for that. We call that hydrolysis. So explosion of water, explosion due to water. Okay. So humans, we need to be able to control, and most, not just humans, but all animals need to be able to control the chemical environment that their cells are in. And so that's why we devote a lot of energy to the homeostatic control of things like pH, so that we can make sure that our cells aren't exposed to environments that have too much salt or too little salt dissolved in it, so that the cell size remains relatively the same. So inside of the cell, when we say that the inside of this, or I'm sorry, these prefixes, hypo, iso, and hyper, are referring to the environment around the cell. So on the left-hand side, hypo means lower or below tonic. So low, below or low pressured solution. So hypotonic solutions have low hydrostatic pressure. The other way to think about that is that they have low salt and low sugar content in the solution relative to the inside of the cell. So if we do a quick example here, okay. So if we say that if we've got our cell here that has a 5% salt concentration, and the environment that it's in, okay, out here, if the environment out here has something like a 2% salt concentration, the solution out here is considered hypotonic because the salt concentration is lower than it is inside the cell. So this would be considered hypotonic. Okay, hypotonic, below what's going on in the cell. Iso means the same, right? So we're seeing here with these arrows is we're showing the net movement of water. In an isotonic solution, the rate of water going into the cell is the same as the rate of water leaving the cell. The only way that that could happen is if you have equal concentrations of salt solution in the external environment relative to the internal environment. So we have what we would say a net zero movement of water. This would be a solution that is at equilibrium, if you want to think about it that way too. So the cells will retain their normal shape as a result of that. But then sometimes you can have solutions that become hypertonic, where the salt concentration is much greater outside the cell than it is inside the cell. So any water that's remaining inside of the cell is going to want to escape because it's moving down its own concentration gradient. Okay, so then the cells will shrivel up, right? Or it's called crenation. You can see down here. So that's important to understand these different solutions and these different chemical environments that cells can be in. Animal cells, human cells, like to be in isotonic solutions, which is why something like our pH of our blood, for example, since this is a blood cell, is roughly around 7.4, right? So it's roughly neutral. So we don't really have too many dissolved solutes floating around in our blood that can mess with the pH in a significant degree, right? And remember, we get around that by having other chemicals called buffers that combat these other chemicals that would cause our pH to change. And therefore, that would change the solution and therefore change the tonic solution of the cell, or of the environment. Okay, so different tonicities: hypotonic, iso, hypertonic. Hypotonic, low salt concentration relative to the inside. Isotonic, the same hypertonic higher salt concentration relative to the cell. Okay. So here we have a good example of something like facilitated diffusion, or this would be, this is a good example of facilitated diffusion because we don't have ATP being involved. We have no energy investment being utilized. So sometimes large molecules like glucose can move through transport proteins that can physically change their shape, but don't require any amount of energy invested. So that's what we have going on here. And we can see, obviously outside the cell, we have 3 glucose molecules. Inside the cell, we have 0. Makes sense that our concentration gradient goes that way into the cell. But sometimes we have these secondary active transport mechanisms that utilize the concentration gradients of one molecule or ion relative to another. The sodium-potassium pump is a great example of that. So for every 3 sodium ions that we invest here, we utilize 1 molecule of ATP to shove all of those sodium ions against their concentration gradient to get out of the cell. Okay, so the concentration gradient of sodium would actually be going into the cell because remember, by definition, active transport shoves things against their concentration gradient, it goes uphill. So the gradient of sodium must be going in so that there's a high concentration of sodium outside relative to the inside. And then we allow potassium to move down its own concentration gradient. So potassium moves into the cell passively once ATP has been utilized. Okay. So this is an example of secondary active transport, where you push one thing against its concentration gradient using ATP, then you allow something else to pass back down its own concentration gradient without the use of energy. You don't need to invest ATP there. And you might be thinking, well, why the heck would our cells do that? If we invest a lot of energy in doing something one way, why would we want to just allow that to be reset? Well, it's kind of silly, but a lot of our cells, that's the way that they function. They work based on shoving things against their own gradient just to allow them to come back in. The mechanism for ATP synthesis is exactly that way. We invest a lot of energy in shoving hydrogen ions against their own gradient just so that we can let them come back into the place that we forced them out of. It's a really cool cycle, and it seems redundant, but it's the best way that our cells have evolved to be able to produce a lot of energy very efficiently. So it may seem dumb at first, but it's actually pretty ingenious if you think about it, because it's a pretty energy efficient loop. So for every sugar molecule, so for every molecule of glucose that we ingest, then we can produce roughly 36 to sometimes 38 ATP molecules just by relying on this cycle. That's pretty cool. So here's a good example of that as to how you can combine or couple different kinds of active transport with different kinds of receptor mediated or carrier mediated transport over here. So if we shove sodium across one side, we can let it come back through with another molecule. This example over here with number 2 is something called a symport protein. So sometimes we can utilize, so sim meaning same, we can allow both of these, so the sodium ion and this molecule of glucose. It's kind of hilarious because the size difference is actually pretty substantial. Glucose is much bigger than just a single ion of sodium here. But if we allow sodium to pass back through, then that changes the shape of this protein to let glucose back through. So this is a good example of something that we call symport transport. Okay, and then this is an example of something that we call receptor mediated or carrier mediated. Actually, I would say for this, it's probably a better instance to use the word receptor, because each of these little molecules out here, these are proteins, but they've got specialized portions of them that respond to concentrations of a specific molecule. So this may be food, this may be some bacteria, we don't know. But each of these proteins will respond to that and trigger a process that has them fold in on themselves like this and create a little vesicle. This is how cells are going to be able to bring in nutrients from their environment to satisfy their own metabolisms. So this is a good example of endocytosis, more specifically receptor-mediated endocytosis. Okay. And then if we want to do the opposite, we just call it exocytosis. So endo, meaning within, cyto, cell, is process, cell within process, and then exo, outside, expulsion of particles, getting rid of waste products. This is the process that we utilize, okay? So the reason that this works is because the vesicles are made up of phospholipids. So when they encounter our cell membrane, they fuse with it very, very well, right? Because they're made out of the same stuff. So it's not like you're adding some new component. It's not like you're trying to mesh wood into steel or anything like that. You wouldn't weld a piece of wood to steel, right? You can weld two pieces of metal together because they're made out of the same stuff, right? I mean, those of you guys who do a lot of welding, you can correct me, on terminology later. But the point is the vesicles are made out of phospholipids. So when they encounter a membrane or another structure that's made out of the same stuff, it's also made out of phospholipids, they fuse with it. This is something that you'll see come up consistently in anatomy, because anytime you have a bit of tissue that globs onto another tissue, if it's made out of the same kind of cells, they will stick together, right? This is why things like your eyeball cells all stick together, and they're separate from the tissue that makes up your eye socket, and they are separate from the bone that makes up your skull, right? All of these different things are made out of different cells, so they all stick with the cells that are similar to them, right? So let's see. So here's what that looks like underneath a transmission electron microscope. So last week we learned about how light microscopes work. So we use light to shine light through a really thinly cut specimen. Transmission electron microscopes utilize the same thing, but instead of photons, they use electrons. And they shoot electrons really quickly through a substance, and we can get really, really high resolution images of things that are very, very small when we do things that way. So what we're actually seeing here, all of this is a silhouette. This is a two-dimensional image. These are silhouettes of particles within a cell that are being expelled through exocytosis, right? But we have another cool bit of technology that we use called scanning electron microscopes, or you'll see it sometimes written as SEM. And that's where we utilize electrons at comparatively low power. So the electrons can't pass through an object, but they bounce off the surface. It operates very similarly to sonar, or kind of like ultrasound, if you guys are familiar with those. And we can get really cool three-dimensional images of cellular and sometimes subcellular compounds. Okay, so let's look into some specific organelles. This is what our nucleus looks like, zoomed in. The nucleus itself has its own phospholipid membrane to isolate it from the rest of the cell. That's important because we have all of our DNA in here, and all of that DNA is what carries our information to look the way that we do. And we are eukaryotes, so we keep all of our DNA compact and inside of the nucleus. But believe it or not, they actually don't exist in a chromosome form until the cell is ready to divide. Most of the time, it looks like you just spilled a plate of mom's spaghetti all over the inside of the nucleus right here. It's just a big spaghetti monster mess for the most part. And so again, another transmission electron microscope here. So we're seeing a two-dimensional image and high resolution of the inside and kind of the outer membrane out here of our nucleus. Right? So we've got a little condensed region of that called the nucleolus, and then we have our nuclear envelope out here, and then chromatin. So in this case, the cell is probably getting close to dividing because we can see these condensed regions of DNA that we call chromatin. So how does DNA get out of the nucleus? Because we utilize different components of the DNA, and I'll correct that. DNA itself doesn't actually exit the nucleus until it's getting ready to divide and it's in a chromosomal form. But we've got a bunch of pores all over the nucleus, and this is that technology I was just talking about, the scanning electron microscope, where this is a three-dimensional image of the surface of the nuclear envelope here. And we've got all these little craters, right? Those are actually pores that open up to the inside of the nucleus. We can release specific components that we generate from the DNA that are called RNA. And we've got a couple different kinds of RNA, but the one that we really care about is something called messenger RNA or mRNA, because that's kind of like the blueprint for making proteins. So here, our molecule of DNA, for the most part, for the most of the life of a cell, will exist in a big, unwound, spirally mess. It'll exist in this double helix form, but it typically won't look like a chromosome until it's getting ready to divide. When the cell when the DNA condenses, it wraps around these specialized proteins that we call histones, and it creates these little, the proteins themselves actually kind of look like kettlebells. If you guys are familiar with what kettlebells look like, that's literally what a histone kind of looks like under a microscope. So we can condense and wrap the DNA around these little clusters of histones to make a condensed form of that DNA called a chromosome, right? And once it starts condensing, that's when we call it chromatin, because it actually is dense enough that we can associate a color with it. So that's where the word chroma comes from. So this figure really kind of summarizes what I've been talking about, especially with respect to getting information out of DNA. The way that I think about DNA is that DNA itself, so like we're seeing here, right? So all these little stringy bits, the DNA is kind of like this super old wise monk who knows literally everything about everything, but he speaks in a language that the average person doesn't really understand. So in order to figure out what the heck this guy's saying, we have translators. We have people that write down what the heck he's talking about. And we call those people transcribers, or in the cellular world, so using our analogy here, those are what we call transcriptors, okay? And the typical transcriptor are these little components in here that read out sections of the DNA, all right? So the rRNA is what typically will read out and transcribe regions of the DNA, write down what this guy is saying, and give that information to people who can interpret what the written word is and spout off all the information that we need to create a fully functional protein. The guys that read out the written transcription are the translators. So we have free-floating ribosomes that are outside the nucleus that translate the information that's written out on the messenger RNA, to the mRNA molecule here. So those two processes together are what we call transcription and then translation. So transcription always occurs in the nucleus where the DNA is, because this is where our super wise monk is. So the transcriptors write out what he's saying in the form of this mRNA transcript. That mRNA transcript leaves the nucleus through the nuclear pores and then encounters ribosomes, which then translate that information into proteins. The rest of the cell speaks in the language of proteins. So if you're not speaking the language of the rest of the cell, then nobody knows what the heck you're talking about. So the translated part right here is going to end up being converted into a protein. So what's going on here with this mRNA that's passing through this ribosome is now adding on little amino acids in a specific sequence that's written out on this little mRNA strip, this little code, right? And it's a smaller code compared to the long length of DNA because we don't need to know every single thing that's going on our strand of DNA. We only need to know specific parts, right? So that's what's being depicted here, is both the process of transcription and translation. So transcription happens first inside the nucleus, translation happens outside the nucleus. Okay, so then once all of that transcribing happens, it can happen either in the cytoplasm on free-floating ribosomes, or it can happen within the rough endoplasmic reticulum where the bound ribosomes are at, okay? And then we've got smooth endoplasmic reticulum over here that doesn't do anything with protein synthesis, but remember can help kind of detoxify some toxins that the cell can encounter, but it also produces lipids that can add on to the structure of the surrounding cell membrane. So another transmission electron micrograph of the rough endoplasmic reticulum over here, and we can see it's kind of hard to tell, but all these little black dots, those are all ribosomes that are stuck into the folds of that folded structure, that endoplasmic reticulum. All right, next up, Golgi body, or sometimes called the Golgi apparatus. Does a lot of the same stuff, but really remember the purpose of the Golgi apparatus is to modify proteins that get made. So all the proteins that get produced from the nucleus or the rough ER or free-floating ribosomes will pass through the Golgi body for the most part, especially if they're proteins that are going to be moving out to the cell membrane. They always have to pass through the Golgi body because the way that they get embedded in the membrane is by being inside the vesicle, which we know is made out of phospholipids. Okay, so here's what that looks like under a transmission electron micrograph. And if you've noticed, like some of the magnification values to 96,000 compared to something like the endoplasmic reticulum, which is at 30,000 times magnification. So really, really small stuff, what we're talking about here. So in terms of size, the Golgi body is obviously going to be much smaller compared to something like, you know, the rough ER, close to 3 times smaller. Okay, so then here's the basic premise for how all of that works, right? So proteins that need to be modified pass through the Golgi body here, but what we're seeing is this is some sort of foreign particle that needs to be broken down and digested. The Golgi body can produce these specialized kind of these aren't the peroxisomes, but they're specialized vesicles that contain digestive enzymes or specialized proteins that can break down other things. So what we're seeing here is the fusion of this lysosome that gets produced by the Golgi body fusing with the vesicle that has some sort of foreign particle, and then those enzymes will break down whatever is inside of this digested vesicle here. Be broken down, and then whatever it is can either be ejected through exocytosis or further metabolized into other things that the cell needs. So let's say, for example, that this was some molecule of glycogen that need to be broken down into glucose molecules, right? So now we've got a bunch of little individual glucose molecules here. Where the heck are those going to go? To our favorite organelle, the mitochondria, right? So the mitochondria is going to be able to process the components of glucose that end up actually getting broken down in the cytoplasm, and then all of that stuff gets moved into the mitochondria where we make all of our ATP. So if you remember, I said that the nucleus contains most of our DNA. Not all, but most, roughly like 99% of our DNA. The other really small percent occurs within the mitochondria. The mitochondria has its own amount of DNA. And there's a whole lot of evolutionary theory that we're pretty sure about that can explain why that happens. And if you guys are curious, we can talk about that later on. But it's significant and it's relevant because when we're developing and when we're growing in utero, all of our mitochondria actually come from our mom. So we're taught that, you know, our genes are roughly a 50-50 split between mom and dad, right? But it's actually 51% mom and 49% dad, because the other 1% comes from all of the DNA in the mitochondria that the egg contains during the fertilization event. So thanks, mom, for all of your mitochondria. Okay. So we can see through a transmission electron micrograph here that the insides of our mitochondria have folded membranes. So these, again, serve the purpose of increasing surface area to volume so that we can make sure that we're maximizing the amount of energy that our mitochondria are producing relative to the amount of space that they take up. Okay, so here we have some of the structure of our cytoskeleton. So we've got a couple different components here. So we've got these things called microfilaments, which are the smallest subunits, roughly 8 nanometers in width. And they have these little small protein subunits that we call actin. And if you guys know a little bit about muscles, you know that actin is one of the major proteins in muscle. The other one would be myosin, which isn't found here. But these actin protein subunits serve to kind of help with resisting tension that can be put on the cell. So if you try and squish a cell or pull on it, the actin filaments serve to resist those pressures. And then the other ones, these microtubules and intermediate filaments here, these serve to help resist compressionary forces. So I know I just said that microfilaments resist compression. That's not right. Microfilaments resist pulling or tension, and then microtubules and intermediate filaments resist compression. So that amount of force, or that kind of force. And here's what those things look like This is more specifically something that we call a centriole that has these little kind of triplets of these protein subunits, these intermediate filaments here. Actually, I'm sorry, it's not intermediate filaments, they're the microtubules. So each of these tubes is a microtubule, one of these things right here. So they get arranged in triplets because it turns out this is a fairly sturdy and stable configuration. So why does this matter? Well, this matters because these are the kind of scaffold mechanisms that can elongate in the cell as the cell is preparing to undergo cell division, and they'll attach to chromosomes and then help provide a track or a pathway to allow chromosomes to migrate from the middle of the cell out towards the poles. And so we can see here that they exist at kind of right angles to one another, which is kind of interesting. And then here, this is just another figure that summarizes, let's see, oh, we're almost done actually, this is good. This figure again summarizes the transcription and translation part of protein development. So just kind of rehashing what we talked about before, DNA gets unwound, we read a little section of it, we make a little blueprint for what we need, that gets sent out into the cell, into the ER or a free-floating ribosome, and then that ribosome takes that little sequence of the messenger RNA and then adds amino acids onto it in a specific order. So these little tRNA molecules, these transfer RNA molecules, are what we get from our diet. So when you talk about like getting amino acids from different kinds of foods, like plants or meats or whatever, this is the form that we consume them in, or the form that they get brought into the cell in these what we call tRNA. Whoever discovered these first thought that they looked like a T, so we call them tRNAs, right? And so then there's a specific process through translation that we end up acquiring amino acids and putting them in chains to make specific proteins, right? So this is important because this is why we're not self-feeding organisms. We don't produce these on our own. So we need to get these amino acids from our diet. We need to get them from different food sources. And certain foods are rich in some, and other foods are rich in others. And so that's why it's important to have a diverse diet to account for that. Let's see. So yeah, really quickly moving through this. This is what our DNA looks like when it gets unwound. We break the DNA apart down through the middle right here. Okay. So this specific figure is actually talking about DNA replication, not necessarily DNA transcription. So when we create a new strand of DNA, we end up adding on a new what we call daughter strand right here, right? And then on the other side that gets split apart, we add in a new strand over here. So a way that we can visualize this if we have the original parental strand of DNA over here, so this is the parental strand. What we're showing here is that once we split it apart, when we make our new molecule of DNA, what ends up happening is half of it acts as the old template or one side of the DNA, and then the other side ends up being made out of a new strand of DNA. So this is what we call the daughter strand. So what we're actually doing is we make two molecules of DNA as a result of this. Let me see. Try and go in reverse here. Okay. So now where we had one molecule of DNA, we end up with two, each one containing half old information and then half new information. And then again, we've got a closer look here at the translation process. So the ribosome has these specific binding sites that will read out the sequence that's depicted on the messenger RNA strand. So each one of these little nucleotides here is put together in a specific sequence because as they get read out, they end up producing specific amino acids. So you can see here they're read out in triplets. The one that's on the mRNA strand right here is the one that we call the codon. Think of it as just like computer code. And then the opposite part is what we call the anticodon. The anticodon is what is read out on the tRNA molecule that contains a specific amino acid. We've got 20 different kinds of amino acids, so there's actually 64 different permutations that you can get, but that we have overlaps and we have duplicates. So we've got 20 different distinct amino acids. So through the process of translation right here, what ends up happening is our starting tRNA molecule kind of shifts sites as the ribbon of mRNA moves through, kind of like a conveyor belt. And then we open up another site to allow for a new tRNA molecule to be brought in. Then what ends up happening is we shove that first amino acid onto our growing chain right here, forming a peptide bond, and therefore creating a amino acid chain. And then this happens over and over and over until we get the designated amount of amino acids in the specific kinds and the specific orders. Then this gets released, this amino acid chain, and then it goes into another molecule that folds it. And then we make our protein, and Bob's your uncle. And it goes off and does whatever it does. And then here is the same thing that I was talking about with DNA replication, where we end up splitting our old strand in half down the middle, and then we add half of a new strand on either side. So when we split the old one in half, we end up making 2 new strands, right? Each one containing half of the old information and then half of the new information, right? Okay, so then finally, this figure highlights cell division. It highlights mitosis. So the important takeaway here is that when we talk about mitosis, we are dividing a cell completely in half, right? So we're making actually I shouldn't say that. We are duplicating the amount of DNA that we have, and we are creating 2 identical daughter cells that have the exact same genetic information in them. So that's the big takeaway, is with mitosis. You go from 1 cell to 2, and they are genetically identical to one another. Same exact DNA that's present in them, right? So the nucleus ends up dissolving. The chromosomes at this point have already, or the DNA has already condensed into a chromosome form. And then they end up lining up along this midline of the cell right here. And the centrioles that I had talked about before, so these things have elongated and are now attached into the middle of our chromosomes here. And then through some other molecular signals, they will pull, or I should say, they don't actually pull the chromosome apart. The chromosome actually has these digestive proteins that will kind of chew away, kind of like Pac-Man. It'll chew itself towards the centriole poles on either side, right here. So then once that happens, then the cell begins to divide. So now we've duplicated our DNA, we are now dividing, and then the cell pinches itself in half, creating 2 new daughter cells that have the exact same genetic information in them. Okay, so that's our end product of mitosis. And this occurs in pretty much every single one of our cells, except for our sex cells, right? So bone cells do this, nerve cells do this, muscle cells, red blood cells. Actually, red blood cells, it's kind of a lie. They don't actually behave in this same way. Red blood cells get broken down and recycled, and then they're made anew within bone. But all of our other cells, everything that has a nucleus is going to be doing these things, okay? So kind of how does this, or how does something like age affect this? Well, age affects this by reproducing, or having cells reproduce at much slower rates, up to the point where they just kind of stop reproducing altogether. Or when they reproduce, they produce cells that are kind of non-functional, or they don't really work the way that they're supposed to. This is why you have instances or rates of cancer are higher in elderly individuals, because as our cells continue to replicate, the DNA gets shorter and shorter, and then you end up printing cells that have misreaded portions of the DNA, and then proteins stop functioning, and then all that other stuff. So pretty much with age, we end up seeing cellular health decline. But what's weird is that even when we're not super old, our cells actually have a pre-programmed lifetime, and then they will actually self-destruct through a mechanism that we call apoptosis, or pre-programmed cell death. Lots of our epithelial cells, for example, have that built in. because our skin cells are constantly reproducing and pushing off all the old dead cells. And so then we slough off all of our old dead skin cells, and then the new ones end up popping up until they die, and then the cycle repeats itself. But yeah, so that's the end of chapter 3. So we finished with a little bit of time to spare. So we're not going to jump into chapter 4 today. We'll leave that for Tuesday. But kind of put yourself in the mindset of thinking like, okay, now we're done with our atomic, chemical, and cellular level of matter hierarchy. Now we got to start thinking about, okay, what happens when we start putting these cells together? What's that going to transform into? Well, we're going to end up getting different kinds of tissues as a result of that. So chapter 4 is going to be all about tissues. All right, I'll stop our recording here.